Welcome to Type 1 Diabetic Warriors. Our team is committed to bringing Type 1 diabetics the latest medical developments to manage this disease. We also cover current medical advancements seeking a cure for T1D. If you value this content, please feel free to show your support by clicking on our homepage and hitting the link to buy us a coffee. Your support helps our team to research topics for upcoming videos. We thank you for your support.
Well, guys, you know, if you have been watching this channel for any amount of time, it is my opinion, I'm not a doctor, but from the research that we do here, it is my opinion that there is not going to be one single therapy that cures type 1 diabetes. I think that the evidence is showing that it is going to be multiple therapies used together that are ultimately going to find the cure for T1D. Now, today we're going to highlight a story about a small molecule therapy that has been designed and is being implemented and is going into trials hopefully next year. And it's all happening at the Cornell School of Medicine, specifically the Weill Cornell Medicine, which is obviously a subsidiary of Cornell University. We have the whole story for you right here, and we're going to share it. And if you would be so nice as to hit the like and subscribe button, that helps us to reach more people with this valuable information. Now let's find out what the story is about this small molecule therapy and how it may very well augment other therapies toward a cure for type 1 diabetes.
Small molecule therapy improves islet transplant success in diabetes, a study from Weill Cornell Medicine. Here's the latest on the study. Weill Cornell Medicine researchers have discovered a new sex-tailored small molecule pretreatment that significantly enhances the survival of transplanted pancreatic islet cells, key to treating type 1 diabetes. Originally a cocktail called LIP, greatly improved islet survival in female mice. However, male mice didn't respond. By adding histamine and serotonin, they developed an enhanced cocktail, LIPS, which worked in male mice too. The team leveraged a high-throughput screening approach, ChemPerturb-seq, coupling chemical screening with single-cell RNA-seq and barcoding. This allowed identification of effective pretreatment combinations at scale and led to a publicly accessible dataset on ChemPerturb-DB featuring an AI assistant. This pretreatment step, administered before transplant, helps preserve islets when transplanted into an accessible site like under the skin instead of into the liver. Ultimately, this innovation could double the yield of donor cells, meaning fewer donors are needed per patient. Why this matters? Enhanced efficiency. One donor could potentially serve 2 recipients. Precision approach. Use of different cocktails for male versus female recipients maximizes cell survival. Open science. ChemPerturb-DB is publicly available, supporting transparency and encouraging follow-up research. What's next? Expanding testing to human islets and diverse preclinical models, advancing toward regulatory approval and clinical trials to evaluate safety and efficacy in humans. Here's a deeper dive into the Weill Cornell Medicine study on small molecule therapy to improve islet transplantation success in type 1 diabetes, focusing on the methodology, mechanisms, and implications. Study overview: improving islet transplant survival. The problem: pancreatic islet transplantation is a promising therapy for T1D, but up to 60% of islets die shortly after transplant due to inflammatory and immune stress. Current delivery into the liver causes additional cell stress and immune exposure. Donor shortages limit widespread application. The innovation: ChemPerturb-seq plus small molecule cocktail. Step 1. High-throughput screening with ChemPerturb-seq. The team developed ChemPerturb-seq, a platform combining the following: small molecule perturbation, apply hundreds of chemical compounds to the islets. Single-cell RNA sequencing, track gene expression responses to each treatment. Barcoding, each condition is traceable at the single-cell level. This allows precise identification of which combinations of preconditioned islets resist stress before transplantation. Think of this as training the islets for survival using targeted molecular tools. Step 2. Discovery of the LIP and LEP-JES cocktails. IGF-1 promotes survival and regeneration. Prostaglandin E2 reduces inflammation and enhances islet function. LIP treatment led to up to 80% islet survival in female mice. Sex-specific response. Male mice did not show a significant benefit from lip. LEPs for male recipients added histamine and serotonin to the lip cocktail. Histamine modulates immune and vascular responses. Serotonin enhances beta cell regeneration and anti-apoptotic signaling. This updated formulation rescued islet survival in male mice, matching lips' efficacy in females. So, how is this therapy delivered to the body? The delivery site. Moving away from the liver, islets were transplanted under the skin subcutaneously instead of into the liver. Less inflammation and hypoxia, more accessible for monitoring or retrieval. Compatible with devices like islet encapsulation or scaffolds. Outcomes. Survival doubled with pretreatment compared to controls. Functional engraftment improved, providing better glycemic control in recipient mice. Potential to halve the number of required donors per patient. What's next? Preclinical testing in human islets. Current results are in murine islets. Trials in human islets are underway using the same cocktails. Combination with other advances. Pairing with immunosuppression-free scaffolds or encapsulation devices like those from Vertex, Cernova, or ViaCyte. Clinical trials pathway. Regulatory toxicology for each molecule is well known. They're all FDA-approved or naturally occurring. Could accelerate translation to first-in-human trials within 1 to 2 years if safety is validated. Significance for type 1 diabetes. This strategy does not attempt to cure autoimmunity but aims to increase the efficiency of islet transplants, reduce donor burden, potentially extend to stem cell-derived islets like those therapies from Vertex, Cernova, and Sana. It acts as a universal enhancement layer, making many current T1D cell therapies more viable. This study supports the concept of using multiple therapies from various pharma companies to ultimately provide the solution and cure for T1D.
Well, what do you guys think of that? I'll tell you, one of the things I found most interesting in this story is that they are going to be working with other technologies that are created by some of the big names who are pursuing a cure for T1D. Sana Biotechnology, of course Vertex, the Cernova Cell Pouch, and of course Evotec that is going to, I guess they call it Evotec. I call it Evotec. Evotec, whatever. They are in the business of mass producing islet cells, and as you know from one of our other stories, they are building a new factory in Portsmouth, New Hampshire to do just that. So the gist of this story is that this is not going to be a cure in itself, but this is going to make the whole idea of transplanting beta cells and curing diabetics much easier. So it will play a role in the process. But think of perhaps the process like 5 fingers on your hand. It may be several different technologies that get to the ultimate goal of finding a cure for this. Whatever the case, we're finding it very interesting. I was never particularly a science geek in my life, but after 42 years as a diabetic, I have to say, since we created this channel, I find this science more and more interesting with every video we do. And I hope you feel the same way. And I hope you feel optimistic, because I certainly do. Now, we'll have another video for you here real soon. We thank you for hanging out today with us here on Type 1 Diabetic Warriors. You take care of those blood sugars, and we will talk again real soon. Have a great day. Bye for now.